Okay, so welcome back to this next video in which we are discussing calcium signaling. Okay, right, so in the previous video what we discussed is the basic overview of what calcium signaling is all about. Okay, what I want to discuss in this next video is normal calcium homeostasis within a cell. Okay, so we discussed that in the resting state before any calcium signaling has occurred, calcium in the cytoplasm is at this very low level. It's at 100 nanomolar, much lower than the extracellular calcium, which is 1.5 millimolar. We also discussed how in the resting state we've got a lot of calcium stored in the endoplasmic reticulum, bound to calcium binding proteins such as calsequestrin. Okay, so what I want to look at in this video is how do we maintain these gradients basically? How do we keep the concentration of calcium in the extracellular fluid higher than the concentration of calcium in the cytoplasm? Or rather, how do we keep cytoplasmic calcium concentration so low? Okay, so there are three transporters that I'm going to talk about which are capable of transporting calcium ions out of the cell basically. Now, one of them is going to be a primary active transporter. It's going to get the energy to do this from the hydrolysis of ATP. The other two are going to be exchangers, which are going to allow other ions to move down their electrochemical gradient, and that will release the energy needed to move the calcium out of the cell. Okay, so let's talk then about these transporters in the cell membrane. Okay, so I'm going to start off by discussing the plasma membrane calcium ATPase, or the PMCA, okay, which initially I'm just going to show as a blob here sitting in the cell membrane. So this is the cell membrane. We'll say this is the extracellular fluid side of the cell membrane here. Okay, and we'll say this is the cytosolic side of the cell membrane here, so this is the cytoplasm. Okay, and sitting in the cell membrane here, we have the plasma membrane calcium ATPase. Okay, now the plasma membrane calcium ATPase, for short, is usually abbreviated down to the PMCA. So this stands for plasma membrane. Whenever you see PM in biology, it usually stands for plasma membrane. Okay, and then the CA is for calcium, that's the C. Okay, and then the A is for ATPase. Okay, and that name instantly tells us that this is going to hydrolyze ATP down to ADP, an inorganic phosphate, and that implies that this is going to be a primary active transporter that's going to get the energy for the movement of calcium out of the cell, this extrusion of calcium from the hydrolysis of ATP. Okay, and indeed that's what PMCA is going to do. It's going to chuck a calcium ion from the cytoplasm into the extracellular fluid, and it's going to get the energy to do so from the hydrolysis of ATP down to ADP, an inorganic phosphate. Okay, now calcium isn't the only thing, however, it moves. It also actually moves two protons back into the cytoplasm of the cell, okay, for every time it moves a calcium out. However, the protons do not have a particularly powerful electrochemical gradient across the cell membrane, so it's not the case that when you move the protons back in that is giving out energy that's needed to move the calcium across. The main source of energy here is coming from the hydrolysis of the ATP here. Okay, right. So, that is one mechanism then for extruding calcium from the cytoplasm of the cell and throwing it out to the outside of the cell, and therefore a mechanism for maintaining a low intracellular calcium concentration of 100 nanomolar. Okay, now I want to discuss the mechanism of PMCA in more detail with you, okay, because it's what is described as a P-type ATPase. Okay, and I want to make sure you understand what is meant by a P-type ATPase, okay, because circa, which we'll see in a moment, which is present on the endoplasmic reticulum membrane, is also going to be a P-type ATPase, okay, so I want to make sure that you understand what is meant by this. So, P-type ATPase, the P means phosphate. Okay, it's standing for a phosphate group. Okay, and we'll see that the addition of a phosphate group is absolutely crucial to how P-type ATPases function. Okay, indeed, the phosphate group is going to cause the conformational change in the plasma membrane calcium ATPase. Okay, so I need to take a step back now. So the key thing to understand about all transporters, whether they are primary active transporters or secondary active transporters, is they have two states, basically. Okay, so let me just draw this here. So now I'm going to draw the PMCA with a more fancy picture, basically. Okay, so here this is now representing my PMCA. So basically PMCA can have two states. One of them is an inwardly facing state. So here the binding site is facing into the cytoplasm of the cell. Okay, and the other state that PMCA can be in is an outwardly facing state where the binding site is facing the outside of the cell, basically. Okay, so this isn't just for PMCA, this isn't just for P-type ATPases, this is for all transporters. This is a very general principle that they have these two states, an inwardly facing state facing the cytoplasm and an outwardly facing state facing the extracellular fluid. Okay, and the key idea is that the PMCA and indeed all transporters need to move from one conformation to the other. Okay, so let me illustrate this point by using this example of PMCA. Okay, right. So PMCA, when it's in the inwardly facing state like this, it's going to bind a calcium ion to its binding site here. So let's just colour this in. So we'll have PMCA once again represented in red here. Okay, and here comes our calcium ion, which I'll underline in blue. So when you're in this inwardly facing state, calcium is going to bind onto the binding site that's facing the cytoplasm of the cell. Okay, then what's going to happen is the transporter is going to change conformation, and I'll discuss exactly what makes it change conformation in a moment. Okay, but we'll leave that bit off for the moment. Okay, it changes conformation to the outwardly facing state here. Okay, and I'll colour this once again in red here. Okay, so it changes conformation to this outwardly facing state. Okay, and in the outwardly facing state, the calcium that is now bound to this binding site, which is now facing the extracellular fluid, is going to cleave off the outwardly facing PMCA now. Okay, so this goes off into the extracellular fluid. Okay, and now the PMCA that is in the outwardly facing state and has no calcium bound to that binding site anymore is now going to bind to protons instead. Okay, so the basic principle is that this binding site of the PMCA in the inwardly facing state has a high affinity for calcium. So calcium likes to bind there, but then when it changes conformation into this outwardly facing state here, the binding site now has a low affinity for calcium, so the calcium gets released, and it has a high affinity for protons, basically. Okay, then the idea is that what can happen is it can change back to the inwardly facing state, and again we'll discuss what causes this movement back to the inwardly facing state here. Okay, that's the bit that's missing in this story at the moment. But here it is now moved back into the inwardly facing state, and when you're in the inwardly facing state, the binding site now has a low affinity for protons, so the two protons get released into the cytoplasm, and of course we're then back to square one, where we can now bind another calcium ion there instead of the protons, okay, because the inwardly facing binding site has a high affinity for calcium ions. Okay, so the portion that we now need to add into this is what actually drives the movement between the inwardly facing conformation and the outwardly facing conformation. Well, that is going to be ATP in the case of PMCA, and in the case of P-type ATPases, which PMCA is, then this movement between the inwardly facing state and the outwardly facing state is driven by phosphorylation by the ATP molecule. Okay, so let's go back here. So the PMCA in the inwardly facing state has bound calcium to this binding site here. Now what's going to happen is to force this into this second conformation, this outwardly facing conformation, you have to phosphorylate the PMCA basically. Okay, So here is this phosphate group that's been added on to PMCA, and it is ATP that's going to do this phosphorylation. So ATP is going to give up its gamma phosphate, okay, and become ADP, and that gamma phosphate is going to be attached onto the PMCA, which I might just label it up here. Okay, so ATP gives its gamma phosphate group onto the PMCA, and that phosphate group that's been added onto the PMCA, that now causes the PMCA to move into the outwardly facing conformation. So here, when it's now in the outwardly facing conformation, it still has that phosphate group attached. Now, once it's in the outwardly facing conformation, the binding site loses affinity for calcium, so the calcium leaves. It gains affinity for the two protons bind, okay, and now you have to move back to the inwardly facing conformation. How does that occur? Well, this phosphate group has to be cleaved off. So basically the phosphate group is removed, and then that causes the movement back to this inwardly facing state, basically. Okay, so when you remove the phosphate group, you go back to being in the inwardly facing state. When you put the phosphate group on, you go to the outwardly facing state. Okay, then once it's moved back into the inwardly facing state, of course, the binding site now loses its affinity for protons, so they move out, okay, and then a calcium moves in. So that's the central principle with P-type ATPases, that the movement between the inwardly and outwardly facing state is driven by phosphorylation. That's why they're called P-type ATPases, because the phosphate group is so utterly crucial to the mechanism by which they move from the inwardly facing state to being in the outwardly facing state. Okay, so that is the mechanism then of PMCA, and indeed it highlights how P-type ATPases work as well. Okay, right, so PMCA is one of these mechanisms for extruding calcium from the cytoplasm and thereby keeping cytoplasmic calcium concentration low. Okay, now there are two other proteins that are also going to be involved in keeping cytoplasmic calcium low, and they are going to be examples of secondary active transporters rather than primary active transporters. So the first one then that I want to talk about is the sodium-calcium exchanger, okay, which I'll just represent as a green blob here. Okay, right, so this is going to be the sodium-calcium exchanger, and for short, the sodium-calcium exchanger is abbreviated to the NCX. So once again, this is the plasma membrane here, this is the extracellular fluid, this is the intracellular fluid here. So NCX stands for sodium, okay, that's the N, so of course we're using the periodic table symbol for sodium, which is Na, okay, then the C is for calcium, okay, and then the X is for exchanger, okay, so this is the sodium-calcium exchanger. Right, and we'll colour in the sodium-calcium exchanger in green here. Okay, right, so the sodium-calcium exchanger then is another way of getting rid of calcium from the intracellular fluid. Okay, so it's going to move calcium from the cytoplasm to the extracellular fluid. Now, where is it going to get the energy to do this? Well, it's not going to get it this time from the hydrolysis of ATP. Okay, so it's not a primary active transporter. Instead, it's going to be a secondary active transporter. It's going to use another ion's electrochemical gradient to harvest energy to move the calcium out of the cell. Okay, so what it's going to do is it's going to move free sodium ions from the extracellular fluid into the intracellular fluid, down sodium's electrochemical gradient. So remember, sodium has a higher concentration extracellularly than it does intracellularly. So I'll just remind you of the general concentrations for sodium extracellularly and intracellularly. So the concentration for sodium extracellularly is usually around 145 millimolar. The concentration of sodium intracellularly is usually around 12 millimolar. So you've gone around a 12-fold concentration gradient for sodium, favouring its movement into the cell. Okay, in addition, you can't just consider the concentration gradient here, you have to also consider the electrical gradient. Okay, when you combine those together, it's called the electrochemical gradient. Okay, so what's the electrical gradient for sodium? Well, let's study the electrical potential difference across the cell membrane. So normally, cells have an electrical potential difference across their cell membrane of around negative 65 millivolts, okay, which means that the intracellular electrical potential is lower than the extracellular electrical potential, and it's lower by around 65 millivolts. Okay, now sodium ions are a positively charged ion. They want to be where the electrical potential is lower, okay, which is currently the intracellular compartment. The intracellular compartment is lower in electrical potential by 65 millivolts than the extracellular compartment. Okay, so sodium ions are going to want to go into the cytoplasm of the cell down their electrical gradient. So that means that the concentration gradient and the electrical gradient for sodium are in agreement. Okay, they are both working together to mean that sodium overall wants to come into the cytoplasm of the cell, basically. Okay, that is the direction in which the electrochemical gradient for sodium is telling it to move into the cell, basically. So, when you move free sodium ions into the cell, that's going to release the energy that you need to move calcium up its electrochemical gradient, because of course calcium is not going to want to move out of the cell. Its concentration is much higher extracellularly than intracellularly. The electrical gradient says go into the cell as well, okay, so calcium doesn't want to move out, so to get the energy to do that, you're having to bring in the free sodium ions. Okay, so that's an example of a secondary active transporter, and we're not going to go into the mechanism by which that works in any more detail, but again, it will work through this basic concept of having an inwardly facing state where it will bind calcium, then an outwardly facing state where it will release the calcium and bind sodium, and then it will move back into the intracellularly facing state where it will release the sodium and rebind calcium. Okay, right, so now the final one that I want to talk about is the sodium-potassium calcium exchanger, or the sodium-calcium-potassium exchanger rather. Okay, so let me put this on here. Okay, so another protein that's involved in extruding calcium is what's called the NCKX, okay, which is the sodium-calcium-potassium exchanger. Okay, so again, we're using the periodic table symbol for sodium, which is Na, then calcium is the C, and then we're using the periodic table symbol for potassium, which is K, okay, and then the X is then again for exchanger. Okay, so this is the sodium, calcium, potassium exchanger, or the NCKX. Okay, and we'll colour in the NCKX here in orange. Okay, so again, this is going to be extruding calcium from the cytoplasm of the cell, and this time it's going to be using the electrochemical gradient of sodium and also potassium to drive this movement. Okay, so this transporter then, again it's a secondary active transporter, and it's going to move a calcium ion out of the cytoplasm of the cell, but along with it, it's also going to move a potassium ion out of the cell. And in exchange for all of that, it's going to move 4 sodium ions into the cytoplasm of the cell. Okay, right. So here, the energy that is needed to move the calcium out of the cytoplasm of the cell, it's obviously being provided by the 4 sodiums that are coming into the cytoplasm of the cell, but it's also being provided by the potassium that's moving out of the cytoplasm of the cell, because potassium's electrochemical gradient is usually just in favour of potassium moving out. Okay, so let me tell you the concentration gradient for potassium. Okay, so the concentration of potassium intracellularly is usually around 155 millimolar. The concentration of potassium extracellularly is usually around 4 millimolar. So it's around a 40-fold gradient usually for potassium, favouring its movement out of the cell. Okay, so a strong concentration gradient saying go out, But of course we have to remember there's the electrical gradient, and potassium is a positively charged ion, so it's going to want to go into the cell because of the electrical gradient. So in the case of potassium, the concentration gradient says one thing, the electrical gradient says the other thing. Okay, who wins is the great question. Okay, and at this electrical potential difference of negative 65 millivolts, it's the concentration gradient that wins. The concentration gradient is stronger than the electrical gradient, meaning that overall the electrochemical gradient for potassium favours its movement out of the cell. You'd have to take the electrical gradient down to the negative 85 millivolts, okay, the Nernst potential for potassium, in order for this electrical gradient to be strong enough to match this concentration gradient. Okay, and obviously negative 65 millivolts isn't strong enough, basically. Okay, so potassium does have an electrochemical gradient favouring its movement out of the cell then. So when we move the potassium out of the cell, that will also release us some energy. Okay, so that's the energy then in this case that's going to drive the movement of calcium out of the cell by these sodium-calcium-potassium exchanges. Okay, right, so we've now seen these 3 different mechanisms then for extruding calcium from the cytoplasm of the cell. Okay, now, they are not all normally functional when resting calcium concentration in the cytoplasm is present. Okay, so basically these two down here, NCX and NCKX, they have an extremely high throughput. Okay, so these two are capable of moving, in the region of around 20,000 calcium ions out every second. Okay, so each one of these pumps can move out 20,000 calcium ions per second, so they have an incredibly high throughput. They can get rid of calcium very, very fast. However, they are not normally functional when the intracellular calcium concentration is normal. Okay, so when intracellular calcium is 100 nanomolar, okay, and extracellular calcium will be 1.5 millimolar, then these are not functioning. Okay, the reason is that they have quite a low affinity for calcium, so they have a high throughput but a low affinity. Okay, so it means that they don't bind calcium very well. Okay, so when you have a low calcium concentration, when you have 100 nanomolar calcium concentration, these things just don't end up binding that much calcium, and therefore they just can't really function at normal calcium concentration. So really, these are off at normal calcium concentration. Contrast that to the plasma membrane calcium ATPase. Okay, the plasma membrane calcium ATPase, because it is a primary active transporter, it is much, much slower than the secondary active transporters. That's a basic principle that goes far wider than just this. Primary active transporters are slow, secondary active transporters are fast. Okay, and indeed the throughput of this PMCA is around 30 calcium ions per second. Okay, so each PMCA protein can pump out around 30 calcium ions per second. Okay, so pretty pathetic compared to the NCX and the NCKX. Okay, however, it has much higher affinity for binding calcium, so it binds calcium much better at a more normal resting calcium concentration in the cytoplasm of the cell. Okay, so it has a higher affinity for calcium. So basically the PMCA is more involved in actually maintaining normal resting calcium level than the NCX and the NCKX. Okay, because little perturbations in the calcium concentration, little rises, okay, tiny little fluxes in the calcium concentration, and the PMCA will deal with it. It will move that calcium out, whereas these two won't, because they need much higher calcium concentrations to actually have a function. Okay, so the PMCA is the one that is dealing with the little day-to-day fluctuations in the intracellular calcium concentration, and then extruding that calcium to maintain the intracellular calcium concentration at 100 nanomolar. So PMCA is the main one maintaining an equilibrium. normally. Okay, so what do the NCX and the NCKX do? Well, they're really important when you actually get a calcium signal and you want to reverse the calcium signal quickly. So if you suddenly do get high calcium in the cytoplasm of the cell because you've had a calcium signal, okay, so because calcium has come in from the extracellular fluid or because it's been released by the endoplasmic reticulum, you want to get that calcium out now of the cytoplasm, okay, quick. You don't want the calcium signal to go on too long. It's meant to be a transient thing. It's supposed to flash and then it's supposed to go, okay. So NCX and NCKX are really important there. So when calcium does go up, they suddenly become functional because now the calcium concentration is high enough that they're actually going to get calcium bound to them at a reasonable rate, okay, and then they have this incredibly high throughput so they can get rid of the calcium very, very quickly. Okay, so they're important in reversing calcium signals basically, whilst the PMCA is important in maintaining normal resting calcium concentration inside the cytoplasm of the cell. Okay, right, so we will call it there for this video, and in the next video we'll continue this discussion by studying the Circa pump, which is on the endoplasmic reticulum membrane, and then we'll move on to store-operated calcium entry, which is the way that you can refill the endoplasmic reticulum store if calcium level in the endoplasmic reticulum store goes down.